All right, Chapter 4, quiz. In a plant species, if the B allele blue flowers and the B allele white flowers are incompletely dominant, so big B, little b is light blue, what offspring ratio is expected in a cross between blue flowered plants and a white flowered plants? So this is a little bit tricky because I'm not following the standard parlance for dominance recessive relationships. So big B, little b. So if we just draw out the genotype table, big B is blue. Big B, little is light blue, and little b is white. So this is an example of incomplete dominance. So no longer is the heterozygote sharing the same phenotype as one of the parents, but now we see a brand new phenotype. And so what offspring ratio is expected in the cross between a blue flowered plant and a white flowered plant? So this is the cross right here. So we know that all of the offspring would be heterozygote. So this individual would provide one gamete, this individual would provide one gamete, and so we know with certainty that the genotype is the heterozygote. So the answer to this one is C. The color of sunflower pollen can be orange, yellow, or white cream. Crosses reveal that true breeding orange is dominant to both yellow and white cream. However, a cross between true breeding white cream and yellow plants produce F1 plants with orange pollen. The F2 populations of this cross suggested result in a ratio of 9 orange, 4 white, and 3 yellow. So what type of inheritance does this suggest? So immediately what you should notice is that these are ratios of 16. So whenever you see 9 to 4 to 3, you just want to add these together, and that equals 16. And so that immediately should force you to think about 2 different genes are involved. So the only way you can get ratios of 16 is if 2 different genes are involved. And so most likely you have a situation like this. This is another thing whenever you think that 2 genes are involved to do. So you want to write out there's 4 main categories of genotypes. So assuming dominance recessive relationships exist between the alleles, then that means that you want to write out the possible genotype combinations like this. So big A dash, big B dash, so that means one allele is big A, and then the second allele can either be big A or little a, and then the same is true for that. And so just in a standard intercross, you would expect to get these genotypes in these ratios. And so, all we're doing is a heterozygote cross like this. And so, this suggests that we have something like this. So 9 to 4 to 3, occurred. And so the way you can get that is that if 2 of these categories, 2 of these genotype categories have the same phenotype. And so this is an example of epistasis. So you should always be thinking about epistasis whenever you see this modification of the 9 to 3 to 3 to 1. And so, the way we can explain this is that this category and this category both share the same phenotype. So does this make sense? So crosses reveal that a true breeding orange is dominant to both yellow and white cream. So let's see how that plays out. So a true breeding orange, that would be this. Cross to a true breeding yellow, that would be this. And so we know that all of the offspring would have that genotype. And so that genotype, according to this relationship, is orange. Okay, that makes sense. So now let's do the next one. So is it orange crossed with white cream? So there's 2 different ways that can occur. So it can either be a true breeding white that is this, or a true breeding white that is this. And so in this case, you would get a, in the other case, you would get this. So again, these would both fall within this category right here. And so if we crossed a orange to a true breeding white, we would also end up with orange individuals, orange pollen individuals. So only when you get to a cross between these individuals and intercross between these individuals do you actually get to see all the different phenotypes. So now we look at the answers, and so we can exclude codominance, incomplete dominance. So this is not an example of having multiple alleles. We would expect to see 1 to 2 to 1 ratios in that case. Dependent assortment, that's kind of a nonsense term. And so now there's 2 different types of epistasis that can explain this. And so is this recessive epistasis or dominant epistasis? So one way to remember this is that chocolate labs, black labs, and yellow labs are an example of recessive epistasis, and they have a 9 to 3 to 4 ratio. So, that's one way you can answer this question. And then the other thing is just, so where does the masking occur? So the masking occurs in this particular category. And so we see that if gene B is recessive, homozygous, then it doesn't matter what A is. And so the categories, so and we can do that using both of these categories right here. So these two categories are both white, and so all that matters to determine whiteness is whether or not B is homozygous recessive. It does not matter what A is anymore. And so this is an example of recessive epistasis, is because if you have a gene that's in the recessive state, it masks the other gene. And so the answer is B. Muscular dystrophy is caused by a recessive X allele, a man with this disorder. So let's look at this. So recessive X-linked. And so a man with this disorder must have the following genotype. So they must have one allele of the muscular dystrophy because this person has it, but we know that he is a man, and so he has received the Y chromosome from the father. So he could not have inherited from either parent because we know that he got the Y allele from his father, and so the answer is B, he must have inherited from his mother. So he cannot pass it along to all of his children because none of his sons will be able to get it. So if he has sons, then he knows that he passed on his Y chromosome. And so he can pass it on along to his sons. No, he can only possibly pass it on to his daughters. And the only way his daughters can get it is if their mother is also a carrier or also has this disorder. You have red guinea pigs, black fur is dominant to white fur. You are surprised when your 2 white guinea pigs have all black F1. When you cross the F1, you observe 9 to 7 black to white. So how do you best explain this? So again, 9 to 7, add that up, and that's 16. So immediately you should be thinking there's 2 genes. And so we're going to go back to this. Let's see if I can just copy and paste this even. We're going to go back to this. No, it's not going to let me do that. All right, so immediately you should think of 2 genes, and immediately you should think of the following genotype categories. And again, there's typically 9 to 3 to 3 to 1 ratios for this. And so we see 9 to 7 to 7, or 9 to 7. And so, if we assign this as such, then we add up the 3 to 3 to 1, so 9 to 7 is possible. So does this explanation answer the question? So you're surprised when 2 white guinea pigs have all black F1. So is this possible? Yes, this is possible if a white individual with the following genotype is crossed with a white individual with the following genotype, then we know these offspring will have this genotype. So they'll be heterozygous, and so if we look at this table up here, this would be black. And then if we intercross this is an intercross between heterozygotes, then we would get the standard kind of 9 to 7 relationship. So this is an example of epistasis, and the best answer for this, so this is not the incomplete dominance or codominance. This is not an example of that. It's not an example of lethal alleles. It's not an example of sex-limited expression. Two genes are responsible for this trait, and they happen to have an epistatic relationship. All right, in the pedigree of the Bombay phenotype, the arrow points to the program and demonstrates that the ABO blood type what? So hopefully you look over at this and your understanding of how the AB system works well enough to know that something is up. So how do we do that? So we know that this individual right here must have this genotype, and I'm not exactly sure how to do subscripts here, but this is an AB blood type, and so this individual must be IAIB. This individual has an A blood type, and so there's two possibilities for this. This individual can either be this or this. And so can they have an offspring that has an O blood type? And the answer is no, because this individual, the mother always passes on either the A allele or the B allele, and since this is dominant over the little I allele, there's no way to have a blood type of this. So we know that the O blood type, if we just follow the standard genes, would be little I. And so the answer to this question is that it is dependent on another gene. And so there's a second gene that can actually also cause you to have O blood type that's not related to the to the I gene. And so the answer to this, which is kind of explicitly gone over in the textbook, we did not cover it in the lecture. The answer to this is that the mother could have been, for example, this, and sorry, the father could have been this, and the mother could have been this. So there's a second gene, and they are both carriers for this, and so they could potentially have offspring that looks like this. And so if they have the recessive allele of the second Bombay gene, then they also have an O blood type. So this is tricky. I will not actually test for the Bombay phenotype on the exam, but I feel like a quiz is a good time to kind of challenge you all with difficult problems. Number 6, 2 mutant lines of flowering plant have been attained, M1 and M2, and each bleeds true for white flower color. A cross of wild type to M1 results in purple color. A cross of wild type to M2 also results in purple color. If the cross of M1 and M2 also results in purple color, what can you conclude about the two lines? So this is a question on complementation. And so this is Drosophila. So we're following the Drosophila nomenclature for alleles. So wild type is ++, and that is purple. Mutant 1 is M1, and that is white, and mutant 2 is M2, and that is white. So the first two crosses just tell you that these alleles are recessive to the wild type allele. So by doing these crosses, you're making either this or this. And you're told that both of those results in purple. So we just know that the wild type allele is dominant to the mutant alleles. And then finally we do a cross of M1 and M2. So now we've created an individual that is heterozygote, and this individual is purple. And so this is an example of complementation. If they are purple, then the genes then the mutants have complemented each other, and so the answer is that they must be in different genes. So if you revert to the wild type phenotype, then that means they must be in different genes because the two mutants complement each other, and there must therefore be the situation where they're in separate genes. So this indicates that this mutant is actually something like this. And so they have wild type alleles of both, and since we know that the wild type allele is dominant, then this individual is purple. All right, number 7, you are told that eye pigmentation of a Martian gene is controlled by a single gene, but there are 5 different alleles. of that gene that can lead to 5 different eye colors. If true breeding black-eyed Martian dog is crossed to a true breeding white-eyed Martian dog, what is the maximum number of eye colors that can be observed? So the answer to this is 3. And I will accept both 3 and 2, although I would say that 3 is a little bit more correct of an answer. So basically what we have is a situation where we have 5 alleles, A1, A2, A3, A4, and A5. And so you were told that a true bleeding black-eyed is crossed to a true breeding white-eyed. So, let's just say that it's like this. So a true breeding black-eyed is crossed to a true breeding white-eyed. What are the maximum eye colors that can be observed? So the first thing you should know is that the F1 all will have the same genotype, and then an intercross will result in, hopefully you have memorized these ratios by now, 1 to 2 to 1. So there's a maximum of 3 genotypic classes. And so the most number of traits that you could observe is 3. And that's in the situation where all 3 of these have different phenotypes. So I did not tell you that that you could assume recessiveness dominance. And so there's no way for you to say that A1, A5 is the heterozygote is the same as either one of the parents, but I can kind of understand why you might have assumed that this dominance recessiveness might exist, and so in that case I'll accept both 3 and 2 for this answer. ABO blood type is determined by 3 alleles. IA is what to I, and so this is just memorization. Recessive, dominant. So AI is dominant to little I. So if you have a heterozygote, you're still an A blood type. In this case, IA and IB, now you have an example of codominant. So codominant, because the blood type is of the heterozygote is both A and B, and so it matches either of the homozygous parents. Which of the following is consistent with a recessive X-linked trait? So X-linked trait. So now we can start to fill in what we know about these individuals. So this is a female, so if she has the trait, she must be this, and this individual must be this, and this individual must be this. So all we're doing is following the fact that this male individual has the trait, so they must have the recessive allele on their X chromosome, and then this female does not have the trait, and so she can either be this to start out with. Now we look at this information right here. Is this consistent with the father's? Yes. So the father has to pass on an XH to the daughter. And then for the mother to get it, for the mother to be able to pass this on, she must be this genotype right here. So the only way that she can have an offspring that has it is that situation right there. So she is heterozygote, so she can have a daughter that does not have the trait. So if she passes on the wild type X chromosome, then the daughter will not have the trait. And the same thing for the son. So the son can also not have the trait if this first allele is actually passed on to them. So this is consistent with a recessive X-linked trait. All right, so now let's look at this next one. So this person has it, so we know that this father has this, and now the mother is this, and the father is this. And then this individual right here is also this. And so this son has the trait, this son does not, so we know that they have different X chromosomes because of it. We know that the father does not have it, but we conclude that the mother is a carrier because in order to have offspring. So this is consistent with the mother not having the trait, and also consistent with the fact that she can pass on the trait to the offspring. And so is it possible that you have one son that has it and one son that does not? Yes, that's true. So in this, the mother passed on one X chromosome to one son, and then a different X chromosome on to the other son. And then in both cases, the father passed on the Y. Can she also have a daughter that does not have it? Yes. So this daughter, however, this daughter can either be a carrier or this daughter can be a non-carrier depending on what the mother passes on, because we know the father passed on an X chromosome that does not have the trait. So A and B both look good. What about the third one? So now we fill in what we know. And so these are the ones that we know for certainty. So we know that the mother has it, so it's recessive, so she must be homozygous. We know the father does not have it, so we know that his X chromosome is normal. And then we look to see, and then we know that the son has it and the son doesn't have it, so one of them must have different ones. So is this possible? No, this is not possible. And the reason this is not possible is because the father must have passed on the Y chromosome to both of the sons, and we know that the mother can only pass on the carrier chromosome, the carrier allele, to both of the sons. And so there's no way for that son to not have it in this case. So a mother that has it can only have sons that have it. And so this is a pedigree that is not consistent with an X-linked trait. And so the answer is D. Finally, the Martian dog example. So what is the maximum number of eye colors that can be observed in the F1 progeny? And so we already actually answered this question earlier. And so we know that all of the F1s are going to be A1, A5. So this is kind of a trick question a little bit, just because it's testing to make sure that you remember that pure breeding parents can only pass on, true breeding parents can only pass on one allele. And so the F1 progeny can only have one possible genotype. And so that means only one phenotype can be observed, one trait can be observed in the F1 progeny. So the answer to this question is 1. All right, that's it.